In this video, we will discuss about effect of engineering CAR-T with bispecific protein on tumor microenvironment. So basically, researchers wanted to check whether their CAR-T with specific bioproteins will be able to increase immune infiltration in the tumor microenvironment or not. So in order to check this hypothesis, they did the immunohistochemistry analysis on the tumors, and in this they have selected the region of interest that are marked by this circle, and then check two things. First one is they check the spatial protein expressions, and they also checked what are the different kind of immune cells that are present in this tumor microenvironment, or there is a change in the tumor microenvironment compositions or not. So the panel B shows the results about the different kind of proteins that they have find out, and they also checked whether the different conditions behave differently or not. So different conditions means they have used this TAG-72 CAR alone, the TAG-72 CAR with anti-PD-L1, TAG-72 CAR with mutated anti-PD-L1 and IL-2, and TAG-72 CAR with anti-PD-L1 and IL-2. And what they are able to see is that each categories get clustered separately, indicating that each categories behave differently. And among those, the TAG-72 CAR with anti-PD-L2 and IL-2 has high amount of this lymphoid lineage, immune checkpoint, and the T cells activations markers compared to the other case. Even with this mutated PD-L1 behaves differently compared to the case where TAG-72 CAR anti-PD-L1 and interleukin 2 case. And once identifying that the most prominent candidate of the CAR-T bispecific protein behaves differently than the other combinations, they wanted to check whether in terms of the different kind of immune cells, what are the changes that they have seen. So when they check it about this CD3, CD4, CD8a, they can see that they are high in the case where they use CAR with anti-PD-L1 interleukin 2, and even the CD28 shows high, and the PD1 is also high, because the more this immune checkpoint, the more immune response is there, the tumors will, or tumor microenvironment will try to make more PD1 to suppress the immune response. And they also checked the Perforin, which is one of the cytokines that are released by the immune cells or the T cells to kill the cancers. They are also statistically significant in the case where they use anti-PD-L1 and IL-2 along with CAR. And similarly, this VISTA, the VISTA plays roles is different. Its job is to suppress the T cells. So it is also high in the our best candidate case. The reason is because the high immune cells are there in the tumors. So the tumor microenvironment is trying to control this high immune system or the high immune infiltrations by releasing more VISTA. And this CD11b, there is no significant changes, and in the case of CD14 also they do not see any significant changes. So but in the CD163, which is a marker for M2 type macrophages and which promote the tumor growth, is significantly lower. And in terms of the dendritic cells, the marker is CD11c. It is statistically significantly higher, and its counterpart, that is MHC-II, is also higher, because dendritic cells works with in coordination with MHC-II. The PD-L1 ligand is also high in the case, in the best possible candidate case, and the reason could be because more more of this, because of the more immune cells infiltrations, the tumors will try to generate more PD-L1 to suppress the activations of the immune systems. And I'm not able to recall this CD11b is marker for which, but and the CD14. You can refer to the gist or the reference paper for more details. Let us continue our discussions about this Ki-67. Ki-67 is the proliferation markers. We can see that proliferation is high in the case in the TAG-72 CAR anti-PD-L1 interleukin 2 is used, and similarly their apoptosis is also high. And the logical reasons could be that because of the high immune response, the TME is try to counter this high immune cells in by generating more tumors. However, it also leads to high apoptosis because of the high infiltrations of the immune response. So once validating the effect on the different kind of immune cells infiltrations in the TME when they use their engineered CAR T cells with bispecific proteins, they also wanted to confirm other things such as the CD8a relations with its co-stimulatory activation factors, the CD28. They can see that its relationship is directly proportional for the case or the best possible candidate case, and similarly this more activated T cells will cause more generations of the VISTA that can suppress the activated T cells. This is also directly proportional. And in terms of the perforin, the role of perforin is to kill the tumor cells, and we can see that with increase in the concentrations of this CD163, there is a decrease in the perforin, and the reason could be CD163 is the marker for this M2 type macrophages. So when the M2 type macrophages increases, then the perforin goes down, because M2 type macrophages is the marker for this tumor associated macrophage. It promotes tumor growth. So when it goes up, the perforin goes down, because the perforin has a role to kill the tumors. Now this MHC-II and CD11c. CD11c is marker for dendritic cells, and MHC-II, and it works through by getting antigen information through MHC-II. So it is also directly proportional. So all these relationships also seems to be in line with the knowledge that is already available in the literatures. So this also validates that the results that they are getting makes biologically makes some biological sense. So this is how they check the effect of the engineering CAR T with bispecific protein. That is, it can generate the anti-PD-L1 and interleukin 2 in the extracellular matrix and affect the tumor microenvironment compositions. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.